Shortly after enzymes were discovered in the early 20th century, scientists began to consider their practical uses. One of the earliest industrial applications of enzymes is in biological washing powders. Washing powders contain a range of enzymes, including amylase, proteases, and lipases. These help break down some of the molecules found in blood, food, and plant-based stains. Enzymes are proteins that catalyze biological reactions without themselves being changed during the process. All enzymes have an active site. This has a complementary shape to that of a specific substrate to which it is able to bind. The formation of an enzyme-substrate complex is crucial to the breakdown of the substrate into products. This investigation compares the effectiveness of washing powders by measuring their ability to break down starch, protein, and oil. Agar plates are used in this experiment. These can be prepared by heating a solid agar powder in water and allowing the mixture to cool slightly before pouring into Petri dishes. For the purposes of this investigation, 3 pairs of agar plates are prepared. This will make it possible for 2 different washing powder solutions, which are named Brand A and Brand B, to be tested. Each agar pair contains an additional substance. One pair contains milk powder, which contains a white powder called casein. The second pair contains starch powder and iodine solution to show the presence of starch. This plate is blue-black in colour. And the final pair contains vegetable oil and a small volume of alkali called sodium hydrogen carbonate and universal indicator solution. Because of its extra liquid content, this plate will contain agar that has a noticeably different consistency from the other 2. Cutting small wells into the plates will provide a space into which the 2 different washing powders can be placed. For this purpose, a cork borer is used. However, to ensure it is aseptic before making contact with the agar, it is first passed through a Bunsen burner flame. 4 wells are made for each washing powder. One of these in the centre will act as the control and is labelled with a C. The other 3 wells will allow for the collection of repeats. The control washing powder solutions are prepared by heating a small volume in a test tube placed in a water bath at 95 degrees centigrade for at least 5 minutes. The high temperature denatures enzymes in the washing powder solutions, which removes the effect of the independent variable in this investigation. A denatured enzyme is unable to catalyze the breakdown of a substrate into products because its active site has changed shape. This prevents the formation of enzyme-substrate complexes. A dropping pipette is used to add the washing powder solutions to 3 of the 4 wells on each side of the plates. Care is taken to ensure that the volume of washing powder added to the wells is just enough to fill them without spilling over the sides. This is repeated twice more for each plate using the same washing powder solution. It is important to ensure that a different pipette is used to transfer the second washing powder solution to avoid cross-contamination. Finally, a sample of each of the boiled washing powder solutions is placed into the control wells of each plate, which have been labelled C. After the washing powder solutions have been put into every well, the plates are then placed into a tray and then stored in a place where they will not be disturbed. After allowing enough time for the plates to incubate, remove them from the tray. The results should show that halos or rings of colour change have taken place around some of the wells containing the washing powder solutions. These regions are called the zone of digestion. To investigate the effectiveness of the two types of washing powder, the diameter of the zone of digestion around the well will be measured. For the starch agar plate, iodine solution has been added. As the starch is gradually broken down by the amylase in the washing powder to simple sugars, the blue-black colour will become colourless. For the protein agar plate, the white milk protein casein is digested by the proteases in the washing powder to form amino acids. This decolorizes the milk. As the oil is broken down into fatty acids and glycerol by the lipases in the washing powder, the fatty acids neutralize the sodium hydrogen carbonate. This causes the indicator to decolorize. The zone of digestion is proportional to the distance from the center of the well to the outer limit of the circle that has changed color or that has been decolorized. Firstly, for the starch agar plate A, measure the diameter of each of the zones of digestion using a ruler and record your results. Then repeat with plate B. Next, measure and record the zones of digestion on the protein agar plates. Finally, measure and record the zones of digestion on the oil agar plates. As would be expected, the control wells show no zone of digestion. This is because the enzymes in the washing powder have been denatured by boiling. Because 3 measurements were taken for each washing powder on each type of plate, a mean for each set of values can be calculated. The collected data can be used to plot 3 bar charts that show the mean diameter of the zone of digestion for each washing powder solution. The results show that both washing powders A and B are very similar at digesting starch, protein, and oil. This experiment showed how the enzyme activity of 2 different biological washing powders can be compared. Biotechnologists can use the results of similar experiments to help optimize the activity of enzymes at lower temperature. This reduces the energy expenditure of washing machines.